Hello, everyone, and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and I'm very excited to share this episode with you. My guest is Kathleen Lutz, VP of the Jackson Laboratories Rare Disease Translational Center, just in time for Rare Disease Day 2024. Rare Disease Day is an observance held on the last day of February to raise awareness for rare diseases and to improve access to treatment and medical representation for individuals with rare diseases and their families. So, Kat, thanks for joining me. I'm so glad you're here today.
Yes, thank you so much for having me.
Good. All right, so let's get started by briefly explaining to our listeners the Jackson Laboratory, or JAX, J-A-X, and then more specifically, the Rare Disease Translational Center. And if you could tell us a little bit about RDTC's mission, that would be great too.
Yeah, I'd be happy to. The Jackson Laboratory is a really unique institution. It's a nonprofit research institution. Our main campus is in Bar Harbor, Maine, but we also have facilities in Connecticut and in California and Japan. So we're really very global in our presence. But it started out really as a very humble research institution, as I mentioned, off the coast of Bar Harbor, Maine. It really focuses on genetics and genomics, and the mission of the lab is to, look at genomic solutions for rare diseases so that we could not only empower the global biomedical community in terms of improving human health, but very specifically understanding how genomics, genetics, and the interplay thereof influence human health. I think it's really unique because it's really been, central to its mission from the start. It was founded in 1929, interestingly enough, and it was based on using mice as model organisms for genetics research. And even to this day, that really is the core and the central component of JAX. In terms of rare diseases, mice make a fantastic model organism for human genetics in general and for human health, but especially for rare diseases, because even from the start, we were looking at rare diseases from mice who had spontaneous mutations in single genes, and we were able to isolate those single genes, identify what they were, and learn a lot about the function of those genes and exactly what they were doing in terms of the role they played in terms of physiology as well as in terms of disease. And I think it's been really interesting as we move forward because so many of the genes in human and mice are conserved. And so this is a really important point. It makes it such that we can study the genes in mice and learn so much about not only the normal state of the genes, but the disease state and how that translates into human diseases, and especially with rare diseases, because so many rare diseases are genetic in their origin, which means very simply that they're caused by a single gene mutation. And so this is really, interesting from a standpoint of biology, because we're now able to look at genes in mice. We're able to genetically engineer precise mutations in those mouse models. Those mutations that exist in patients that we've known about for a very, very long time, genetic defects like muscular dystrophy, cystic fibrosis, for example, but then also de novo mutations that are much more rare than what you would find in some of the common inherited rare diseases. These are often de novo mutations that occur in the germline of the parents or very early on in embryonic development. And so the mouse really makes for a fantastic model organism so that we can learn a lot more about what's happening in terms of the pathophysiology, again, the disease state of the gene, as well as the normal function of that gene.
Good. Okay, thank you. Let's talk a little bit more about rare diseases specifically. Mm-hmm. And so, a lot of our listeners certainly do have a great deal of, professional experience, academic experience, both in the study of rare disease. What are the specific qualifications of a rare disease?
Yeah, that's a good question. I think for rare diseases, there's a very specific definition in the United States with an incidence of less than 200,000 patients affected at any one given time. I think there's a definition there in terms of, how we focus on regulatory components, but I think rare diseases, as they affect the global population of over 350 million people, are not so rare when you think about it in those terms. And certainly, if you are a parent of a child with a rare disease or have a rare disease yourself, I think the numbers sort of fade into the distance, don't they? They become less about the numbers and less about the statistics, and more about the challenge that's in front of these families. And I think the Rare Disease Translational Center at JAX, we really put a lot of effort behind that because we completely appreciate how difficult it is to have a rare disease. A rare disease is not something that is not necessarily going to be the focus of a lot of pharmaceutical and biotechnology companies, because there's just simply not enough of a population for those companies, right, to invest in. And for better or for worse, I think our drug discovery system is really set up in that regard. No company, there's not going to be a company that's going to get rich off of a weight loss drug or an Ozempic, type of, component. But nonetheless, even if there's only 100 or 200 people who have a rare disease, collectively, it's a very large population of people. And I think when it's your family member or your child or you, I don't think the numbers really matter anymore, right? I think that just because you have a rare disease doesn't mean that you don't have the right to treatment and you don't have the right to the research, because we have so much to offer in terms of what we can do in terms of research. There's therapeutics that are being developed, and if they can be developed for a common disease, why can't we develop them for rare disease? And I think the RDTC, the Rare Disease Translational Center at JAX, really that's our mission, to not discriminate, for lack of a better word, on the incidence of the population, but really try to use the science that we have, the therapeutic developments, the potential for a lot of these genetic therapies to help as many people as we possibly can.
Good. Okay, great. I want to talk a little bit about, how and when the study of a specific rare disease starts. And maybe that differs from disease to disease. Could you give us a little bit of insight around kind of the how and the when?
Yeah, I think, it's an interesting historical perspective, looking back at it. A lot of rare diseases were named after the physician or the clinician who identified or first described that particular disease. And again, depending on how common that rare disease is, for, again, lack of a better word, the incidence in the population would make it such that it was more common and could be diagnosed relatively easily. Before we had genome sequencing, exome sequencing, whole genome sequencing, a lot of rare diseases were not even described. They may have been categorized as looking similar to or resembling a particular disease. Maybe it was lack of reaching particular milestones, learning disabilities, cognition problems with speech. Maybe they were weaker, hypotonia, not growing as quickly. And so the categorization of neurodegenerative, neurodevelopmental, all of those types of big blanket terms really started to pop up. But as sequencing became more and more prominent, and I still think, I'll put a plug in here, that I don't think it's prominent enough. I think we need to be sequencing patients earlier so that we can understand exactly what their genetic mutation is. And that's what we do with whole genome sequencing or exome sequencing. Instead of trying to really identify the disease based on what it looks like, we identify the disease based on the genetic mutation itself. So if we can do that sequencing early enough, we can understand them. Most of the time, I'm going to say a large majority of the time, we can identify not only the defective gene, but right down to the base pair, exactly what's wrong with that child. And that's incredibly powerful because now you have these subcategories of diseases, some that are, genes in the similar pathway, genes that, sort of converge, in different areas. But I think the precision involved in identifying that patient mutation is incredibly powerful. And that's where a lot of the genetic therapies are really geared towards. And it's important because I think it's not a case anymore that you simply say that, your child, has a developmental or a neurodegenerative condition. You're not really sure what it is, but there's some, maybe occupational therapy or physical therapy. And in many instances, as many of us know, most children who are diagnosed with a rare disease don't live past their fifth birthday. And that's really devastating, for parents, for families to have that level of a diagnosis. The idea of being that helpless and not even understanding what's wrong with your child, those days are really in the rearview mirror for us. And I think we have a lot to offer families with rare diseases if we can just get to that genetic mutation, if we can get to the molecular diagnosis sooner rather than later. We really don't want kids to be so far into their disease before we figure out, what the issues are. The sooner we can diagnose a rare disease, I think the better chances and more shots on goal that we have for treating it.
Sure. That makes a lot of sense. Tell us a little bit about how RDTC is using gene therapy as a potential cure for rare diseases.
Yeah, I think this is really exciting. When we talk about rare diseases, and I think about, over the years, even as I've been studying genetics, genomics, and looking, it's one thing to be able to identify a gene that's causative for a disease. It's another thing to genetically engineer that particular mutation in a mouse model so that we can, really study and understand. I mean, the science is incredible. What you can do with genetic engineering, the things we learn, the biology, the technology of omics, and it just seems like this mountain of data and information that is almost a mountain that's hard to climb because there is, so much of it. But I think that the technology of genetic therapies is the most promising and the most exciting. You can't use the gene therapies in isolation. You do have to understand a little bit about that data, what that gene is doing, where it's expressed, how it's functioning. Is there too little, too much? What's actually the nature of the mutation itself? But the gene therapies, I think, are the most exciting because, before we had these gene-based therapies, we were trying to treat, I'm going to say symptoms almost, right? We were trying to get maybe small molecules or drugs that might act upstream or downstream of the genetic mutation, things that could alleviate the symptoms. But when you think about genetic therapies, they really are attacking it right at the core. The gene is defective, fix the gene that's defective. It sounds that simple, and it sounds so simple, and it is that simple, but it's not as simple as it sounds. We still have a lot of work to do. I think the potential for gene therapies is still really, fantastically exciting. I think we do need to catch up. It's lagging behind a little bit, the application of these amazing therapeutics, that we have, and the potential for them needs to really, sort of move a little bit faster because we have all of this research on one end, and all of these potential therapeutics, gene-based especially, that are so incredibly exciting. But on the other side, we have too few, in my opinion, therapeutics that have been FDA approved to treat rare diseases. And we really need to close that gap.
Yeah, sure, absolutely. Now, certainly we spent this time talking about rare diseases and, how far we've come, how far we still have to go. How can the study of rare diseases potentially advance progress in more common diseases?
Yeah, I think we get that question a lot. And I think, I always think about it in terms of the genetic mutations that we know in the common arena, if you will. We know risk to Alzheimer's. We know BRCA1 genetic mutations for breast cancer. We know that there are genetic mutations that have susceptibility to high cholesterol, to blood pressure. Some of these are single gene, but most of them are not. They're more than one gene, and they're complex. But we do know that there are susceptibility genes. And so think about if we could fix a gene for a rare mutation. And when I say fix, loss of function, you can do gene augmentation, gene replacement. Editing is a fantastic strategy. And when you think of it, it's really sort of, it blows your mind a little bit to think of, 3 billion base pairs in the human genome and an editing strategy that can just hone in on that one particular mutation, correct that mutation, and then for all cells that, then therefore divide after are completely rectified. So that's like an amazing, it almost sounds like science fiction, but believe me, it's not. We've been working with base editing in mouse models. We've shown the accuracy, we've shown the specificity, we've shown, the therapeutic potential is really great. Delivery is something that we have to work on because, especially for neurological diseases, hitting the central nervous system is not as easy. But I think that those technological advances will also come. But in terms of common diseases, the therapeutics themselves, I think, are going to first be validated in very severe rare diseases. We know for a lot of these diseases that these kids don't have much time. The clock is ticking. And that creates a sense of urgency. It creates this, I think it lowers the bar a little bit for the FDA. I think we definitely need to be safe. Nobody wants to be cavalier about throwing therapeutics out there that haven't been tested for safety and haven't, been shown to necessarily have the potential to work. So we do have to demonstrate that there is safety. We have to demonstrate efficacy. But in many of these cases, we already know if we do nothing that child will die. And so the bar, if you will, is a little bit lower to move in with these therapeutics because we have to give these kids a fighting chance. We just do. When death is absolutely an option at the end of their disease, intervening is something that we can do. So this is where I think the therapeutics will be validated in rare diseases because of that sense of urgency. We will very quickly, I think, understand the potential for a lot of these therapeutics. And then that will have, ramifications for more common diseases down the line. And I use Alzheimer's and breast cancer because we know that there are susceptibility loci there that are very common in the population. And we know what they are. We know the genes and we know the mutations that render somebody, susceptible to Alzheimer's. And then I think, as we have rare diseases, so not just the genetic therapies, but those things that can work. I think epilepsy is a great example. Things like congestive heart failure are another. If you can find small molecules that intervene and work their ways within those pathways to prevent something in a rare disease, you may also be able to use it for like an intractable epilepsy, for example. So I think there's discoveries to be made on both ends, genetic therapies and small molecules that interplay in those pathways. And I think that a lot of what we learn about common diseases and therapeutics for common diseases will absolutely come from all the work we're doing in rare diseases today.
Yeah, yeah, absolutely. Absolutely. Sure sounds it for sure. All right, I want to talk a little bit about collaboration with RDTC. And from what I understand, the RDTC engages with institutes, foundations, and biotech and therapeutic development groups for preclinical research up and running based on specific patient genetics. And so talk to our audience a little bit about the benefit of collaboration and how can perhaps even listeners, or a qualifying organization get involved with RDTC?
Yeah, that's a great question because I think a lot of times we do see, especially families who are recently diagnosed, finally tackling that hill in terms of, now I understand what the genetic mutation is in my child, and now you're on top of that hill, and now you're facing another mountain of what do I do about it, right? And because the sequencing has been so fast and so quick in its development, I think the number of rare diseases, the number of causative mutations is really piling up, and in some ways it's really exciting because it's a wealth of information, and then in other times we do have to have the research to catch up to all of these discoveries. So NIH has been done a fantastic job. In my opinion, we've been fortunate. The Rare Disease Translational Center, we have a very large grant from NIH that funds the Jack Center for Precision Genetics, and so this is a grant where we're able to take a diagnosis from a patient who comes to us and says, "My child has just been diagnosed with a mutation in this gene." And those dollars allow us to genetically engineer those mouse models, understand really what's happening at the level of the pathology. Because a lot of times these rare diseases have such few patients, it's often hard to even get the natural history together. If it's a very heterogeneous disease, what are we really looking at? If some kids have epilepsies, some don't, some kids are verbal, some are not. So even a single monogenetic disease can be very complex, in some ways. And so having the dollars and having the ability to work with foundations and patients to genetically engineer those models, because that's a big deal, right? Having a model organism for a particular genetic disease gets you so far. It allows you to understand the pathophysiology, the biology. But most importantly, it allows you to have a therapeutic testing platform for things that, like gene editing, gene therapy, viral-based vectors, lipid nanoparticles, all of those things that are so exciting and in development, are now sort of this little patient avatar in the form of a mouse, that can help you understand. And I think, we're no different, I think, than the way science is being done all over, but I think we're exceptionally good at knowing that we are not going to solve these diseases by ourselves. There's no lab who really does. So, one of the things that we do is collaborate. We're not going to be experts in every single disease, although we're pretty good at most things. And I think the one thing that we do, I think, exceptionally well is when we have these mouse models, we make them available almost immediately to anybody who wants to use them, because we may have a very good idea of what we think, we could do, the next experiment, the next therapeutic, but I guarantee you, there's 100 other people out there, who are thinking just along the same ways and have different expertise or have different thoughts or different ideas. And if we can all work together and have those resources available, then these therapeutics for rare diseases are going to move that much faster. So it's been, really exciting to see it come together over the years. It used to be, one gene, one disease, one laboratory, back in the 80s and the early 90s. And certainly if you look at how science is done today, it's done by groups, consortiums, and everybody putting, their best foot forward, and the thing that they're the expert in, into the pot, and then you mix it all together and you come up with, success much faster than you would if you were to try to do this by yourself.
100%. I think that outlook of collaboration is incredibly important for, not only what you're doing, but over, cell and gene therapy, which what we cover as a whole, but certainly for what the RDTC is doing. And, the whole is greater than the sum of its parts. All right, listeners, that wraps up this episode of Cell and Gene, the podcast, featuring the RDTC's Kat Lutz. Kat, thank you so much for your time and for all of the great information you shared with us today. This was fantastic, and we'll hope to have you on sometime soon to bring us up to speed on what you're doing, even, in addition to what is being announced and talked about on Rare Disease Day.
And please, for the listeners out there, go to our website, look at the Rare Disease Translational Center at JAX, see how we can help. We specialize, I think, in newly diagnosed families and individuals, trying to help them get started, trying to help leverage the resources at NIH, the resources at the Jackson Laboratory, and all of the people that we know who are working in rare diseases, to really get things started much more quickly than maybe in years past. So I think it's a great resource for families, foundations, and not only for newly diagnosed, but for those diseases that have more potential therapeutics out there that haven't been tried, for the more common rare diseases and the common diseases themselves. I think the genomics and genetics component of JAX is incredibly special and really primed, for this kind of research. So we're really excited we're excited to be in rare diseases at a time where the therapeutics are just so incredibly interesting and tangible. And right. So helping these patients and these families, I think, is really our mission.
Excellent. Thank you. Well, you heard her. Please go visit those sites and to learn more and to see how you can get involved and learn all that you can learn. And thanks for tuning in, and we will talk to you soon.